Hello class, this is Mr. Roth here, and today I'm going to be teaching you about the cell cycle and about how cancer develops and what cancer is. So first I want to just remind you of the last lesson that I taught you was on cell division, specifically mitosis, which is the typical type of cell division in the body. It's the type of division that makes 99.9% of our body's cells, and it makes an exact clone. And so when we look here at cell number 1 and we compare that to cell 6, which is really close actually to being split into two new cells. Once these two new cells develop, it's important to know that they're exact clones of that original cell. So this is, again, mitosis is again the process by which we do maintenance and we replace the cells that are getting old and dying in our bodies. Unfortunately, however, this type of cell division is not always perfectly regulated. And the problem is, if it doesn't get perfectly regulated, you can end up with uncontrollable cell division, which can end up in a mass and a clump of cells forming in your body called a tumor. And of course we associate tumors with cancer, and when we get these clumps of cells that are dividing uncontrollably, they can cause tremendous damage to our organs and ultimately lead to the death of the organism. Now, it's actually, when you think about it, quite frankly, it's amazing that our cells know how and when to divide in the right times and the right amounts in our body. It's amazing that we don't have more tumors than we do. However, cancer is, of course, a devastating condition. So here is this is what we call the cell cycle, and I want to let you know that this cell cycle indicated here, this is representing mitosis, okay? And it's dividing up the cell's cycle and showing you how much the cell spends, how much time the cell spends in interphase compared to mitosis. And so I want to show you this little sliver here. It looks about like a quarter of the cell's life is actually spent in mitosis, whereas the rest is spent in interphase, which is the cell just kind of growing and hanging out. Now, interphase, this purple phase here, is actually subdivided into smaller portions, and right after a cell divides, so if we go back to this slide, right after this cell splits, each of these cells will have its own life, and it will go on and each of them will start growing. And that initial growth phase just after mitosis comes to a halt, you can think of everything moving in a clockwise fashion here. And of course it's a cycle, so it goes around and around and around over and over again. But after it's just finished mitosis, it begins its growth phase, and we call this the G1 phase. And this is where the cell is growing in size, and it's also making copies of its organelles. So the number of mitochondria are increasing. The number of vacuoles and lysosomes and ribosomes are duplicating and increasing. Now, as that is going on, eventually we get to a phase where the cell is going to replicate its DNA and make copies of the chromosomes. And of course we know that this needs to happen because we're going to split the cell in half. And we know that for a cell to be healthy and fulfill all of its roles and be able to function normally, it's got to have a full set of 23 pairs of chromosomes, totaling 46 total chromosomes. And so we're going to have to duplicate those so that we can divide. That's what happens in the S phase. Okay? And S actually short stands for synthesis. After the DNA has been copied and duplicated, we then move into growth phase 2, G2, and this is going to be where the cell makes its final preparations for mitosis. So it creates things like microtubules and it arranges the centrioles in order to rip apart the chromosomes and bring them to the poles and go through those classic processes of mitosis. After G2, of course, we then come around and we move into mitosis. First phase, prophase, prometaphase, metaphase, anaphase, and telophase. We're not going to go into the details of those, but of course you know those are the phases of mitosis. And then once those are finished, we go into cytokinesis, which is where the cell splits, as I showed you here. And then we have 2 brand new cells. Each of those cells then begins the process over again, and it goes around and around and around and around. Now, as I said before, there are times where things aren't working quite right. And so I want to show you up here, it says checkpoints. Malfunctioning checkpoints are the cause of cancer. And so if we look at this diagram, this is the same as the diagram before. It's just got some extra details, but you'll see that in red are the checkpoints. And so I want to highlight that there is a G1 checkpoint. So as the cell is growing and multiplying its organelles, there comes a point where the cell needs to check for some things. So at the G1 checkpoint, we check for cell size, we check for nutrients, growth factors, and we also, most importantly, we check for DNA damage. We don't want this cell to go and duplicate itself if it's got mutated DNA. That would contribute to cancer. That would be a bad thing. And so this checkpoint is actually, in some sense, a cancer-preventing checkpoint. If it finds that it has DNA damage at this checkpoint, it's actually going to halt the rest of the process and prevent the cell from replicating itself. So there's also a checkpoint at G2, and I wanted to show you that. This is checking for cell size, and it's also checking to make sure, okay, has the DNA replicated? If it has, and it's free of errors, then green light. You get to move on to mitosis and divide. And then within mitosis, there is yet another checkpoint that's checking to make sure that spindle assembly has been created and that the chromosomes are correctly attached. If that's not there, we're not going to be able to very efficiently and effectively separate the chromosomes, and we might end up actually taking 2 chromosomes over here and 0 migrating to the other cell, which can result in a chromosomal disorder like trisomy 21, which is where one of those cells during the division process of meiosis ended up with 3 copies instead of 2. And when you have 3 copies of chromosome 21, that leads to what we know as Down syndrome. Okay, so let's move on here and look at this. I don't know if I've told you this actually, but cyclins they're a very special type of protein that are responsible for controlling the checkpoints, because you probably ask yourself, okay, great, there's a checkpoint. Who or what is actually doing the checking? The answer to that is that these cyclin proteins are designed to as switches. Really, they kind of turn on or off the process, so they can halt the process if they need to. And you'll notice that cyclin G1 is produced initially in the G1 phase. The S cyclins are in their highest concentration right around the S phase. And so you see that these cyclins, they do get created and released at kind of specific times. Now, this is a complex diagram, but I just wanted to highlight that these cyclins, are these boxes here, so cyclin B, cyclin A, as I said before, and they are associated with different parts of the cell cycle. And you see them binding to certain kinases is what these are called. And they have a particular interaction there that's very, very important that we're going to talk about in just a second. And again, just to remind you, what we're talking about here is the regulation of these checkpoints. And we're also talking about when they fail to regulate and we develop cancer. So how exactly does this play out? How exactly do these work? Well, there is something called a CDK, and it's a cyclin-dependent kinase. Its job is to go and phosphorylate things, and when it phosphorylates things, it activates them, turns the switch on. Well, as the name suggests, it is in fact cyclin dependent, which means that if a cyclin, the correct cyclin, is not present, then this particular CDK is actually inactive, as it is shown here. And you'll see there's a little slot here, a binding site for the cyclin, but unfortunately the cyclin's not there. And so the cyclin-dependent kinase does not phosphorylate its targets. And so the S phase factors are in the off position. However, let's say that we are entering into the G1 or approaching the S phase. At that time, as we said before, the G1S cyclin is at its peak, and it is in fact floating around the cytoplasm. And so, if it's floating around the cytoplasm, it floats in and happens to attach to that cyclin-dependent kinase. So when the cyclin is there, it activates the kinase, which then goes and phosphorylates the targets, turning them on and activating them. So when the targets are activated, those targets then go and initiate the S phase. So in some sense, this is the green light here. This is the mechanism by which the green light gets turned on, telling the cell to move into S phase and move forth with the process of cell division. So this is kind of the mechanism by which the cell division is controlled. If something in here, however, gets messed up, you could see how we could lose control of the cell cycle, which of course is what leads ultimately to cancer. Okay, let's look at another example. There is a particular gene that creates a protein called the p53 protein. And it's often referred to, it's a layman's term, as the guardian of the genome, because p53 is responsible for checking for DNA damage. And so if DNA damage is present, p53 gets activated, and it will go and initiate the CDK inhibitor. Okay? And so when DNA damage is present, p53 gets produced. p53 then activates the inhibitor, which goes and blocks the G1 phase from going forward. Now, unfortunately, there's a lot of research suggesting that there's correlational studies that show that the chances of a person who has cancer also having a mutated p53 gene are pretty high. In other words, it's very common for people who have cancer to also have a mutated p53 gene, which means that their p53 protein is, if it's mutated, it's misshaped and it's not going to effectively check for the DNA damage. It might not even effectively bind to the CDK inhibitor, and therefore it's not going to be able to stop the cell cycle as it's supposed to. And so what can happen is, if there's DNA damage, p53 is mutated and can do nothing to stop it. And that's in the example of a person who has cancer. Now, in a healthy individual, if that cell is found to have DNA damage, p53 will detect it and will halt the cell cycle and cause it to pause in the G1 phase, preventing it from going into the S phase, which is where the cell goes and copies its DNA. And this is a good thing because, again, we don't want to copy mutated DNA. I wanted to give a even simpler example here. There are also studies that have shown that individuals who have cancer frequently have mutations in their tumor suppressor genes. And so, as the name suggests, a tumor suppressor gene creates a protein that inhibits the cell cycle. So maybe you can picture this gene down here. This is a piece of DNA, this long strand down here. And on this side, on the right side, is where the gene is located that initiates cell division, let's just say. Well, a tumor suppressor, if it's doing its job, will come in and bind, and here it is, this big purple square. It's blocking the RNA polymerase from moving forward and synthesizing the protein that's going to initiate cell division. And so it's blocking that process from happening. In other words, it is suppressing cell division in this case. Now, unfortunately, those who have cancer or are prone to cancer often have a mutated tumor suppressor gene. So it's the same gene as this one here on the left, but for them there is a mutation located right here, that little red band. And that mutation ultimately leads to a defective and misshaped tumor suppressor. And so you can picture this red tumor suppressor trying to come over here and bind just like the normal repressor, but because it's misshapen, it doesn't connect, it doesn't bind, and therefore it does not block the RNA polymerase. And the RNA polymerase is free to move forward and translate the RNA into a protein, and cell division moves on. And so you can see that a misshapen tumor suppressor fails to inhibit cell division. And if you fail to inhibit cell division, you have unregulated cell division, which leads to tumors and which leads to growing masses of cells inside the body, which of course, as we said before, damage the body and damage our organs and can lead to death. Okay, so that is it for the explanation here. I recommend watching the video 1, 2, 3 times if you need to hear this stuff again, because it is complicated and a bit abstract. I hope this does give you a starting point to begin to understand how cancer develops, and also maybe gets you thinking about, I could maybe think of some cures here for cancer. If I could just fix this, we could prevent cancer. If you got some good ideas, go do cancer research. All right, that is it. We will see you tomorrow. Have a good one. Bye-bye.